Welcome everyone, Dr. Linden here. Today is the third lab. We're going to be covering the 4 types of tissues today. And hopefully today's a little bit of a review, only because I kind of changed the sequence with you this term. I wanted to make sure that you had more time to study today's information. Typically, when we do tissues, I cover the 4 types of tissues: the epithelium, connective, muscle, and nervous tissue. And within that, we cover the bone histology, the red blood cells, the skin, and that's a lot. That's a lot of content to cover in one lab. So what we've covered so far, let's see if we can go through this to make sure we're all on the same page. I want to make sure I didn't miss anything. So we did the introductory PowerPoint within our first lab. We did go over the microscope. We went over the cell model with you, going over the organelles, their structure, their function. Here we did the anatomical position. We did all the reference terminology. We did planes. We did proximal, distal, superior, inferior, medial, lateral, anterior, posterior, right? We did like frontal, mental, otic, ocular, nasal, buccal, cervical, all the terminology, right? Should sound familiar. Beautiful. We went over not only the cell, but we went over cell division, right? PMAT, prophase, metaphase, anaphase, telophase. We showed the 6-minute video going over the sequence of events of cell division. We did go over the bone histology model. And with that, the terms like osteon should sound familiar. Concentric lamellae. Remember, that looks like the tree that was chopped down and it has all those concentric rings. Yes or yes? Yes, good. So we did the concentric lamellae. We did the interstitial lamellae, the lacunae, the cavity, the canaliculi. We spoke about the difference between compact bone and spongy bone, where compact bone has the osteon, with mentioning the main bone cells like the osteoblast and the osteoclast. We talked about bone remodeling, where osteoblasts build up the matrix of bone and then osteoclasts break it down. And when they're in good ratio to one another, when osteoblasts are building up bone at the same rate that osteoclasts are breaking down bone, that's healthy. But if you have an accelerated rate of which osteoclasts are breaking down bone faster than osteoblasts can build up bone, that's osteoporosis. Okay, just trying to put some content to why you're learning this stuff. We did talk about 3 types of muscle, right? Skeletal, smooth, and cardiac. I'll go over those again today when I go over the 4 types of tissues, but this time showing it under a microscope slide, so you'll see them. We talked about the blood cells. We differentiated between red blood cells and white blood cells, or erythrocytes and leukocytes. Under the white blood cell, the leukocytes, we broke them up into granular sites and agranular sites. On the granular sites, we spoke about BEN, B-E-N, basophil, eosinophil, and neutrophils. On the agranular sites, where the cytoplasm did not have any granules, those were the lymphocytes and monocytes. And we showed you what they look like on those slides, as well as the plaque. So we did cover that. What I'm going to do today is we're going to cover the 4 types of tissues. We're going to review them, spending, when I say review, we're going to review the muscle. That will be review. Under connective tissue, there's many categories under connective tissue. Bone is one, blood is one. So in that respect, it'll be review, but we'll see other types of subcategories under connective tissue. Epithelial will be new, so we'll talk about epithelium, connective, muscle, and nerve. Those are the 4 types of tissues. Now, once we finish the tissues, we'll get into the skin model. And the skin makes the perfect organ to look at first in terms of systems, the integumentary system, the skin falling under that, because the skin contains all four types of tissues: epithelium, connective, muscle, and nerve. Okay, and that's all in terms of content. I will cover that, and then I will cover, we'll go over this skin slide, the skin model here, and I will go over to PowerPoints. One of them is the four types of tissues. We'll go through this PowerPoint. And I will also go through the histology slides. And this will look mostly like microscope slides. And I'll go through these and explain what we're looking at and what type of questions we need to ask ourselves to identify, hey, are we looking at epithelium or are we looking at connective tissue? Okay, so it's really only two PowerPoints that we're going to be looking at today. All right, any questions in general before I get into the content of the classification of tissues? Okay. All right, so let's take a look, and let me enlarge this a bit here. Okay, so to review, I know I've repeated this several times, but repetition works. There are four primary tissue types. There's epithelium, there's connective tissue, and these two are big groups. There's a lot of subcategories under them. Neural tissue and muscle tissue, easy to learn, easy to understand. Not that epithelium and connective is impossible. There's just lots of subcategories that I need to explain to you. So when we look at the classification of tissues, they're all a little bit different, right? I mean, their structure different, their patterns are different. And if their structures and their form or function, if their structures and form are different, then we know that if structure is different, their functions are going to be different. Pretty simple in terms of principles. Remember, cells come together to make up tissues, and then tissues come together and they make up organs. When we study tissues, we call this histology, right? When we study cells, we call it cytology. Now, we're going to look at epithelial first, okay? When we look at epithelium, one of the key differences between epithelial tissue and connective tissue is how they look under a microscope. When we look at epithelial tissue under a microscope, we're going to see things that look like this. How we have a bunch of cells packed closely together. Once you look under a microscope and you see in any one of these cells packed closely together with little extracellular matrix, which means little space and substance between the cells, that's epithelium. Connective is the opposite, right? Connective tissue, we saw a little bit last week where we may have a red blood cell here, and then we may have a large, monocyte here, and then we have a platelet maybe here. So connective tissue is when you have cells that aren't on top of each other, that are further apart away, but there's lots of extracellular matrix and stuff between the cells. That's connective tissue. Epithelium, we have cells packed closely together with little extracellular matrix. That is a really important concept to get into your mind. Okay, so let's go back here and now look at epithelium. So epithelium is the type of tissue that is considered a lining type of tissue or a covering type of tissue. So when we think of something that's covering the external surface of the body, we should think of the skin, but the epidermis, okay? The epidermis. Now, epi means above. So epidermis is the layer that's above the dermis. So epidermis is superficial, the dermis is deeper. When we talk about the skin today, we will discuss the 5 microscopic layers of the epidermis, and they all start with the term stratum. S-T-R-A-T-U-M. They all have the term stratum, or like stratosphere, which means layer, stratum. Epithelium also lines body cavities and tubules. So I'll give you an example. This is a tube, right? This is a tube, a tubule. And we have tubules throughout the entire body. Arteries are tubules, veins are tubules, the esophagus is a tubule, the trachea is a tubule, the small and large intestine are tubules. Anything that makes up a tubular-like system is considered a tubule, okay? And it also lines body cavities. Now, if I were to turn this and change it from like this angle here, and we look at it, let's say we look at this cap, and I turn it from this view to this view, you can see the inside. The inside is what they call the lumen. So if I were to rotate this, now we're looking at it like this from the inside. And something may be traveling here in the center. So that's the lumen. And when we talk about epithelium, we're going to be looking at the type of tissue or cells that line or come into contact with this lumen right here. That's how we identify the type of tissues, is what do we see lining the tubules closest to the cavity or the lumen. All right. Glands are also a type of epithelium. Glands. So we'll talk about certain glands like the sweat gland or the mammary gland. Now, the main function of epithelium, a big important one, is protection. Think about the skin, right? It's protecting us from excessive friction, from bacterial invasion, from chemical damage. If a chemical touches your skin, it's not going to burn and permeate right through muscles and tendons and bones, because we have many, many layers of skin protecting it. But imagine your skin was only 1 cell layer thick. That means you would scratch yourself and those cells would be gone and you'd be seeing what's deeper under the skin. So thank goodness we have many, many layers of epidermis and dermis to protect us. Ciliated epithelium, remember cilia, these are found in the respiratory tract, and they are sweeping dust and foreign particles away from the lung. So if, let's say, here is the airway, and we have some columnar cells here, they look like columns, right? So they're called columnar cells. We're going to have cilia, these little hairs coming off of it. Okay, so this is called ciliated epithelium. And let's say right here, I'm making a type of round cell. And over here, I'm going to make a round cell. These are called goblet cells. And what goblet cells do is they produce mucus. Okay, so this goblet cell is going to release mucus. The way it releases mucus, it's called exocytosis. We remember that term. So it's going to produce mucus, it's going to release it, and it then is going to stick to the cilia. Same thing here. This goblet cell produces mucus, and it sticks to the cilia here. And now those cilia are beating rhythmically in an upward direction, whereas when we inhale air, air comes down this way. So if there's dust particles and debris, the dust and debris is going to stick to the mucus. And then the mucus is moving all the debris upward because that's the way the cilia are beating. The problems with smoking is that smoking paralyzes the cilia and they burn them off. And this is when people smoke for a long period of time, they get up with that smoker's cough, where they get that purulent, producing cough where they have to cough really hard to push whatever's deeper in out. Why? Because they've done the damage to the cilia. And what ends up happening is something called metaplasia. Metaplasia is when one cell or type of tissues die and the body replaces it with something else, where it maladapts, where you're doing damage to the body, it changes it forever and then changes into something else. So these ciliated columnar cells that should normally be there now get burned off, and the body starts to make a cell called squamous cells. And if you ever heard of a type of cancer called squamous cell carcinoma, this can happen with smokers as well. All right, so I'm going to go over this again, where we get to see these cells, what are columnar cells and squamous cells and what they look like. So protection is really important. Let's not harm our natural body's anatomy and physiology, okay? Absorption. So there is special absorptive epithelium. We find them in the stomach and we find them in the small intestine. Now think about it. The small intestine and the stomach is where all the food and nutrients are. So we need to have type of epithelium that can absorb and act like a sponge to pull in nutrients to fuel the body. Epithelium can act as a filter, especially around the kidneys and inside the kidney tubules. There's the word tubules again. The kidneys are not only filtering, they can absorb certain things and they can secrete things from the body. So different types of epithelium that line the kidney tubules, depending on what part of the tubules we find it, it can filter, it can absorb and say, hey, we don't need this, let's get rid of it. So that's kind of neat. All right, and that's why we see here excretion and secretion, right? Different ways of the body getting rid of things. Glands secrete, just like we saw the goblet cell up here secrete mucus. Excretion means getting rid of something from the body. The epithelium also serves as sensory reception, right? So we have epithelium that covers the body surface like the skin, and within the skin we have sensory organs that give you information about, hey, maybe there's an insect that's crawling on you or a mosquito that's on you. It allows you to tell the difference between light touch and deep touch. We talked about the cilia before. We mentioned that they are long, slender extensions. They come off the cells or the plasma membrane, and they help to move secretions and fluids across the cell's surface. That's what we saw along the trachea with the columnar, with the ciliated columnar, and those cilia beating rhythmically upwards. Women also have cilia in your uterine tubes or the fallopian tubes, and they're beating rhythmically to bring the sperm and the egg together for conception. Okay, so we just don't see it along the respiratory tract. We'll see it along the reproductive tract as well. Microvilli, these are smaller projections, and their primary function is for absorption. And these are found throughout the small intestine. So I'm going to draw a simplistic picture here just to give you an idea. So here's part of the small intestine. Whoops. Here's part of the small intestine. And throughout the small intestine, we're going to have a bunch of these microvilli. It'll be like this. You'll have a columnar cell, and then on top of it, a bunch of these microscopic extensions that are much smaller than cilia. And it gives it they call it like a brush border appearance because it looks like the end of a broomstick, many, many filaments at the end. So they call it a brush border. So we're going to have these columnar cells all throughout the intestines, right? And you're going to have a bunch of little microvilli coming off of them. And what's the function of these microvilli? The function of them is to absorb. They're going to act like a sponge and pull in carbohydrates and proteins and fats. They're going to pull in all the nutrients. And then deep, deep here are going to be arteries and veins and lymphatics, and they're going to pull the nutrients into the circulatory system. Okay, so it's going to act as like a connection. The arterioles and the venules and the lymphatics, they're going to transfer the absorption of whatever nutrients were taken in by the microvilli into the cells and eventually into circulation, so that the circulatory system is bringing all of these vital nutrients to all the cells of your body for function. Okay. So now let's look at how do we classify epithelium? What are the questions we need to ask ourselves so when it shows up on a quiz or an exam, you can name it? So when we look at something under a microscope, the very first question we have to ask ourselves is, one of the things before, do these cells look like they're closely packed together or are they far away from one another? And if they're closely packed, we know it's epithelium. So that's the first question. Once we know it's epithelium, then we say, all right, how many layers do I see that are at the apical surface or the top surface or the surface closest to the lumen? Does it look like it's one cell layer thick, or does it look like it's multiple layers? Once we establish that, the first name of the epithelium is either going to be simple or it's going to be stratified. Once again, if we see that it's one cell layer thick, it's simple. And if we see more than one cell layer, it's stratified. Okay. The next question is we would ask ourselves is, all right, what's the shape? What's the shape of the cell? Does it look flat and irregular? If it's flat and irregular, we call it squamous. If it looks like a cube or a square, like this with a nucleus, it's cuboidal. And if it looks like a building or a tall column with a nucleus, then we call it columnar. All right. So let's take a look at the picture. So here we see at the apical surface, this is one cell layer thick. We see this is one cell layer thick. And we see this is one cell layer thick from top to bottom. So all of these have the term simple associated with it. But if we look down here, here is the apical surface, that's the top layer. And we see like one layer, we see two, we see three, about maybe 4 layers here. So because they're more than one layer, the first word is going to be stratified. Here, we see 1, 2 layers, so we call it stratified. Okay. Here in the middle, top middle, we see that these are squares. So because they're cube-shaped, it earns the term simple cuboidal. If we look down at this one, here we have we have 2 layers of cubed. So because they're 2 layers, we call it stratified cuboidal. On the top left, we see these flat, irregular shaped cells. They're not cubes, they're not columns, they're just flat and irregular. So that's the term squamous. And because it's multiple layers, then we call it stratified squamous. Okay? Now, look at the one to the lower right. This one here. We see the word pseudo stratified columnar. Now, the shape of the cells are definitely columns. They're not flat. They're tall and they're slender and they're longer. So those are columnar. But are they truly, truly stratified? Do they look like this where they are clearly more than one cell layer? Or does it have the illusion that it's more than one cell layer? Well, here's one cell and you see the nucleus. Here's another one. There's its nucleus. So if you look at the columnar cells, they really are just one cell layer, but the nuclei are at several layers. So it gives the illusion or the appearance that it is a stratified layer. So this we call pseudostratified columnar. If it was truly a stratified columnar, then we would see something that looks like this. Here's a nucleus, and here's a nucleus, and here, and you can see that it's stacked on top of each other. So that to the right here, this would be really a stratified columnar. So pseudo-stratified gives it the illusion that it is. Okay, and the last type is something called transitional. Transitional, we're going to find around organs that can be, think of a balloon, right? If a balloon is deflated compared to inflated, and you were to draw a number or a shape on a deflated balloon, and then you blow, 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 and you blow it up, all of a sudden the shape changes. Whatever you drew on the deflated balloon alters. It doesn't look the same anymore. So you can have one type of cell that when the organ is distended, like let's say the urinary bladder, when there's urine in it, compared to when someone goes to the bathroom and evacuates, now there's no more urine in there, the cells are going to alter their shape and they go through transitions, right? They go through different shapes and sizes. That's called transitional. Typically, you're going to see them in the urinary bladder and the ureters that attach to it. Okay, let's go back here. So to review, we have epithelium that can either be one cell layer or more. If it's one, it's simple. If it's more, it's stratified. And then we have the flat, irregular shaped squamous cell. We have the cuboidal and the columnar. The squamous cell is irregularly shaped. The cuboid is cube shaped. And the columnar is like building shaped or column shaped. The squamous cells, we're going to find these where we have diffusion taking place, where we need, let's say, oxygen or CO2 to pass through that membrane. So in the lungs, in the deepest part of the lungs where we have alveoli and you breathe in oxygen, oxygen is going to pass through one cell layer membrane, right? If this is oxygen, it's easier for the oxygen to diffuse and get to this side if it's only one cell layer thick, compared to squamous cells that are stratified, that are multiple. It's going to have a hard time getting through a stratified squamous. Okay, so squamous cells are also used for protection. Would you agree that simple squamous is a better idea for protection, or would stratified squamous be better for protection? Well, when we talk about protection, having more layers is going to make more sense than just one cell layer. So we can see squamous as either simple or stratified. The same thing is true with cuboidal cells. We can see them as simple or stratified. Cuboidal cells are found primarily around the kidney tubules. in AMP2, in Anatomy and Physiology 2, when you go over the kidney tubules, you will learn that the functional part, the functional unit of the kidney is called a nephron. Nephron. And the nephron has proximal and distal, there's the word proximal and distal, has proximal and distal convoluted tubules. Convoluted means it really is like all convoluted, right? It's all coiled up and mixed up. So where the proximal and distal convoluted tubules are, we're going to find simple cuboidal cells, and they're used for secretion and absorption. And what the kidneys do are different than what the lungs do. So we're going to have different structure, different function. Columnar cells, we're going to find these in the respiratory tract, like I showed you before in the trachea with the cilia. And we're also going to find them throughout the digestive tract in the small intestines, especially. Okay, so we went over pseudo stratified, right? There's the word pseudo, it actually means false. And we said that the nuclei are at different levels, so it gives it this false appearance that it's stratified. And often where we find pseudo stratified, it's going to be ciliated, especially in the respiratory tract. We're going to see that. We'll even see it in the digestive tract as well. Transitional, which is a type of stratified squamous that can alter in its shape and size, especially if that organ is being stretched. So we'll find it in the urinary bladder, the ureters, and even parts of the urethra. Okay, the cells may appear flattened when the organ is full, and it may look round when the organ is empty. So it's altering and changing its shape, so we call it transitional. So the three types of simple epithelium are simple squamous, simple cuboidal, and simple columnar. Stratified could be stratified squamous, stratified cuboidal, and stratified columnar. And let's not forget about our two unique ones, the false stratified, known as pseudo stratified columnar, and then your transitional epithelium. Okay. All right, so that is epithelial tissue. Okay, I'm going to pause here, and then we'll get into connective tissue. Okay.